Hello friends, welcome to my YouTube channel, Gajpal A Learnings. In this video we are going to study about Introduction to Cellular Level of Organization, which is a part of B Pharmacy first semester, of subject Human Anatomy and Physiology Part 1. Our agenda in this video is to study about Introduction to Cellular Level of Organization, Structure and Function of Cells, Transport across cell membrane, and Cell Division. So let's start. Going towards our first topic, let's see what the cellular level of organization means. At the cellular level of organization, the human body is composed of individual cells, which are the basic units of life. Cells vary in size, shape, and function, but they all share some fundamental characteristics and components. Cells communicate with each other through various signaling mechanisms, such as chemical signals and electrical impulses, to coordinate their activities and maintain overall homeostasis, which means the body's internal balance. They also undergo processes like growth, division means mitosis, and programmed cell death means apoptosis to ensure proper development and tissue maintenance. The cellular level of organization is essential, because all higher levels of organization, including tissues, organs, and systems, rely on the interactions and functions of individual cells. By studying cellular biology, scientists and medical professionals gain insights into the mechanisms underlying cellular processes and can better understand diseases, develop new treatments, and advance our knowledge of human health and development. Now looking forward to the structure and function of cells. The structure and function of a cell are closely linked, as each component of the cell has a specific role in carrying out essential functions necessary for the cell's survival and overall function. Here are some key structures and their functions within a typical eukaryotic cell. First one, cell membrane, also known as plasma membrane. Structure of cell membrane. The cell membrane is a thin, flexible barrier that surrounds the cell, consisting of a phospholipids bilayer with embedded proteins. Function of cell membrane. It regulates the movement of substances into and out of the cell, maintains cell integrity, and provides protection and support. Second one, nucleus. Structure of nucleus. The nucleus is typically a spherical organelle, located in the center of the cell, enclosed by a double membrane called the nuclear envelope. It contains chromatin, DNA and associated proteins, and a nucleolus. Function of nucleus. The nucleus stores the cell's genetic material DNA, controls gene expression, and directs cellular activities by synthesizing RNA and producing ribosomes. Third is mitochondria. Structure of mitochondria. Mitochondria are double-membraned organelles, with an inner membrane folded into structures called cristae. They contain their own DNA and ribosomes. Function of mitochondria. Mitochondria are the primary sites of cellular respiration, where they generate energy in the form of adenosine triphosphate ATP, through the oxidation of glucose and other fuel molecules. Fourth is endoplasmic reticulum, AR. Structure of AR. The AR is a network of interconnected tubules and flattened sacs, extending throughout the cytoplasm. There are two types, rough AR with ribosomes, and smooth AR without ribosomes. Function of AR. The rough AR is involved in protein synthesis, folding, and modification, while the smooth AR participates in lipid metabolism, detoxification of drugs, and storage of calcium ions. Fifth is Golgi apparatus. Structure of Golgi apparatus. The Golgi apparatus consists of a stack of flattened membranous sacs called cisternae. Function of Golgi apparatus. It processes, modifies, sorts, and packages proteins and lipids into vesicles for transport to their specific destinations within or outside the cell. Sixth is lysosomes. Structure of lysosome. Lysosomes are membrane-bound sacs containing digestive enzymes. Function of lysosome. They break down and recycle cellular waste materials, foreign substances, and damaged organelles through the process of hydrolysis. Seventh is ribosomes. Structure of ribosomes. Ribosomes are small complexes of ribonucleic acid RNA and proteins, existing either freely in the cytoplasm or attached to the rough earth. Function of ribosomes. Ribosomes are responsible for protein synthesis by decoding messenger RNA mRNA and assembling amino acids into polypeptide chains. Eighth is cytoskeleton. Structure of cytoskeleton. The cytoskeleton is a network of protein filaments including microtubules, microfilaments, actin filaments, and intermediate filaments. Function. It provides structural support, maintains cell shape, facilitates cell movement. Ninth is nucleolus. Structure of nucleolus. The nucleolus is a dense region within the nucleus. Function of nucleolus. It produces and assembles ribosomes, which are essential for protein synthesis, 10th is cytoplasm. Structure of cytoplasm. The cytoplasm is a jelly-like substance composed of water, ions, proteins, and organelles. Function of cytoplasm. It serves as a medium for cellular processes and houses various organelles. 11th is peroxisms. Structure of peroxisms. Peroxisms are membrane-bound organelles containing enzymes. Function of peroxism. They participate in detoxification processes and break down fatty acids and other molecules. 12th is centrosome. Structure of centrosome. The centrosome contains two centrioles and surrounding pericentriolar material. Function of centrosome. It aids in cell division by organizing the mitotic spindle and ensuring proper chromosome segregation. The coordinated activities and interactions of these cellular components are essential for the proper functioning of cells and, consequently, the overall functioning of the organism. Here is the brief diagram of human cells anatomy with all the necessary structure involved. Now our next topic is transport across cell membrane. The cell membrane, also known as the plasma membrane, plays a vital role in regulating the movement of substances into and out of the cell. It consists of a phospholipid bilayer with embedded proteins that selectively control the transport of molecules and ions. There are two primary methods by which substances can cross the cell membrane: passive transport and active transport. First, passive transport. Passive transport does not require the expenditure of energy by the cell. It occurs spontaneously, driven by the concentration gradient of the substances being transported. There are three main types of passive transport. A. Diffusion. In diffusion, molecules move from an area of higher concentration to an area of lower concentration until equilibrium is reached. Small, nonpolar molecules like oxygen and carbon dioxide can freely diffuse across the lipid bilayer of the cell membrane. B. Facilitated diffusion. Some molecules, such as large or charged molecules, cannot diffuse directly through the lipid bilayer. In facilitated diffusion, specific transport proteins called carrier proteins or channel proteins assist in the passage of these substances across the membrane. This process still occurs along the concentration gradient and does not require energy. C. Osmosis. Osmosis is the diffusion of water molecules across a selectively permeable membrane. Water moves from an area of lower solute concentration or hypertonic to an area of higher solute concentration or hypertonic until equilibrium is reached. The movement of water is facilitated by aquaporins, which are specialized channel proteins. Like oxygen and carbon dioxide can freely diffuse across the lipid bilayer of the cell membrane. B. Facilitated diffusion. Some molecules, such as large or charged molecules, cannot diffuse directly through the lipid bilayer. In facilitated diffusion, specific transport proteins called carrier proteins or channel proteins assist in the passage of these substances across the membrane. This process still occurs along the concentration gradient and does not require energy. C. Osmosis. Osmosis is the diffusion of water molecules across a selectively permeable membrane. Water moves from an area of lower solute concentration or hypertonic to an area of higher solute concentration or hypertonic until equilibrium is reached. The movement of water is facilitated by aquaporins, which are specialized channel proteins. Cotransporter in the intestinal cells, which transports glucose into the cell against its concentration gradient by utilizing the sodium gradient established by the sodium-potassium pump. In addition to these mechanisms, cells also utilize processes like endocytosis, which means taking in substances by engulfing them with the cell membrane, and exocytosis, which means releasing substances by fusing vesicles with the cell membrane. In short, we can say that the transport across the cell membrane is crucial for maintaining cell homeostasis, allowing nutrients to enter the cell, waste products to exit, and ensuring the proper balance of ions and molecules. The specific transport mechanisms employed by a cell depend on the size, charge, and concentration of the substances involved, as well as the energy requirements of the transport process. Cotransporter in the intestinal cells, which transports glucose into the cell against its concentration gradient by utilizing the sodium gradient established by the sodium-potassium pump. In addition to these mechanisms, cells also utilize processes like endocytosis, which means taking in substances by engulfing them with the cell membrane, and exocytosis, which means releasing substances by fusing vesicles with the cell membrane. In short, we can say that the transport across the cell membrane is crucial for maintaining cell homeostasis, allowing nutrients to enter the cell, waste products to exit, and ensuring the proper balance of ions and molecules. The specific transport mechanisms employed by a cell depend on the size, charge, and concentration of the substances involved, as well as the energy requirements of the transport process. Forms visible chromosomes. The nuclear membrane begins to disintegrate, and the centrosomes, containing centrioles, move to opposite poles of the cell. Spindle fibers start to form. C. Prometaphase. The nuclear envelope fully breaks down, and the spindle fibers attach to the chromosomes at specific structures called kinetochores. The chromosomes are now fully condensed and visible as distinct structures. D. Metaphase. The chromosomes align along the equator of the cell, forming a metaphase plate. The spindle fibers are attached to the kinetochores of each chromosome, ready for separation. A. Anaphase. The spindle fibers contract, causing the sister chromatids of each chromosome to separate and move toward opposite poles of the cell. Each chromatid is now considered an individual chromosome. F. Telophase. The separated chromosomes reach the opposite poles of the cell. Nuclear envelopes form around each set of chromosomes, and the chromosomes begin to decondense. The spindle fibers disassemble. G. Cytokinesis. This is the final stage of mitosis, where the cytoplasm divides, leading to the formation of two separate daughter cells. In animal cells, a cleavage furrow forms and pinches the cell membrane inward, while in plant cells, a cell plate forms to separate the daughter cells. This is the diagrammatic representation of mitosis cell division. You can go through it. Second one is meiosis. Meiosis is a specialized form of cell division that occurs in reproductive cells, that is, gametes, to produce haploid cells with unique genetic combinations. Meiosis involves two rounds of division, known as meiosis I and meiosis II, resulting in the production of 4 daughter cells. The stages of meiosis are similar to those of mitosis, but with some key differences. A. Prophase I. This is the longest and most complex stage of meiosis. Chromosomes condense, homologous chromosomes pair up to form tetrads, and crossing over occurs, leading to genetic recombination. B. Metaphase I. Tetrads align at the metaphase plate, and spindle fibers attach to the kinetochores of each homologous chromosome. C. Anaphase I. Homologous chromosomes separate and move to opposite poles of the cell, reducing the chromosome number from diploid to haploid. D. Telophase I. Nuclear envelopes form around each set of chromosomes, and cytokinesis occurs, resulting in the formation of 2 daughter cells. A. Meiosis II. This is similar to mitosis and involves the division of the haploid cells produced in meiosis I. The stages prophase II, metaphase II, anaphase II, telophase II are similar to their counterparts in mitosis. The end result of meiosis is the production of 4 haploid. And now this is the diagrammatic representation of meiosis. You can go through it. Thanks for watching this video. In the next video, we will continue this topic.